thanks very much for everyone for being here today. I'm thrilled to be working with Louise for the past year and a half. And so today I'm going to tell you a bit more about our work on autism genomics. Sorry, is that okay? Yeah. So what is genomics? Perhaps you've probably heard about genomics on the news. Maybe you've heard about ancestry DNA tests where you can, for €100, you can figure out your background testing through DNA. You might have heard the American novelist Dani Shapiro and Ray Darcy recently talking about how she did a DNA test and found out that her father wasn't her father. Or you might have heard about using genomics to track down cases of murderers back in America who 20 years later, they use DNA testing and ancestry DNA testing using genomics technologies. So these are the kind of things that are in the news about genomics that you might have heard about already. You might have read about why genetic tests matter for autistic people on the news. You might have seen Eva on the RTE news there. Recently, we'll have a talk from Eva later on today talking about life-changing genomics treatments. So it's quite often in the news, but it's maybe not clear what genomics is. And then also you might have heard about Generation Genome, where over in the United Kingdom in the NHS, their chief medical officer, Professor Dame Sally Davis, she has said, Generation Genome is here. The age of precision medicine is now, and we have to act fast, and this technology has the potential to change medicine forever. You know how genomic medicine has implications for understanding and treatment of rare diseases, and we can tailor treatment for more patients than ever before, and that patients with a rare disease should have access to genomics-based care and health and care from professionals, and this should be or could be a standard part of the approach. But this isn't in Ireland yet, and genomics has a lot of advancement to do. So what is genomics? It's the study of our genome. And what is our genome? It's all of our genetic code. So Louise, you may remember this slide from Louise earlier on. She introduced genes to us. So our body is made up of cells, and within our cells we have our chromosomes, and then our chromosomes are made up of DNA, and then part of our DNA is, within the DNA is our genes. And just to give you a little more detail, I suppose, to expand more on Louise's figure, is what makes up this DNA, our genetic code. So we have chemicals, a part of our chemicals, and they are labeled, we call them letters, A, T, G, and C. So we've four different types, and we've got 3 billion of these letters. So we've got 3 billion of these genetic code in our genome. So that's a huge amount of data that's compacted into our DNA. And so the study of genomics is the study of this complete set of our genetic information. So the genome is the complete genetic information of an organism, and it includes both your genes and the non-coding DNA. And this is different from genetics, because genetics is the study of heritability. It's the study of function and composition of just single genes, of which we have about 20,000 genes anyway. So it's still very complex. And Louise has talked already about how the gene-specific sequence of our DNA codes for a functional molecule. So how our genome differs, my genome differs from each of you here by about 3 million differences in our DNA, but that's a very tiny amount compared to the 3 billion base pairs that we have. And it's trying to figure out what those differences mean and why I'm different than the rest of you, and how those differences in our DNA relate to how we are. And so where does it all fit in? So this human genetic variation between us all leads to differences in how genes make proteins and how these work in a biological pathway, and we'll hear more about biological pathways from Jamie later on, and how these pathways work together to change the function and work the brain, and then how this comes about into complex diseases. And it's really trying to understand the brain and more all these processes that lead to the brain. And so this is a picture here about how we investigate the brain, different types of biomarkers that we use beyond genetics. And Amber here from Cambridge is going to tell us more about the types of biomarkers they use for studying autism, and genetics is a major part of that study as well. So I guess back to genomics. Here's a picture of me here. You'll meet Niamh later on. She's going to record the event today through her artwork. So she's very talented, and we're hoping that you guys can take part as well if you like outside, and if any of your artists do take part. But we're really keen to illustrate, I suppose, genomics, and I'm going to tell you a bit about the research project that I've recently started within Louise's group. So it's the title is my team's task is discovery to clinical utility of rare genetic mutations in neurodevelopmental disorders. And I suppose I normally like to introduce it as ABC. So A for autism is a predominantly genetic disorder. And Louise already has talked about family studies, twin studies that research has shown suggest the importance of genetics for autism. But I thought this was really interesting. It was a large study in the US of 160,000 families, and they looked at the heritability of 149 different disorders recently. And you see it's ordered into those that are the most highly heritable are at the top here, and it goes all the way back to the least heritable disorder. And those in red are neuropsychiatric disorders, and the top two is autism and ADHD. So it really illustrates the importance of genetics in understanding autism. So, and of course then is the importance of studying families. So we're recruiting and studying families with brothers and sisters and multiply affected individuals within the family. Louise already introduced pedigrees to you, so you can see here that we're interested in families of multiply affected individuals, and hopefully we're doing genome sequencing analysis. So C is for sequencing or CNV detection, which Louise already introduced to you. The apple is just to give you an illustration of the amount of data, the amount of coverage, the amount of technology that we're using. So when we talk about sequencing, we talk about measuring each part of the 3 billion base pairs that we have within our DNA. And you can do this in so many ways. And very recently, we've been able to measure each of the 3 billion base pairs within our DNA. So that's like capturing the whole apple. But you can also just measure, say, people think the important bits, the genes within them. So that's called the exome, which is the slice of the apple there. That's 1%, and that's about 20,000 genes. Or you can also, within those 20,000 genes, only look at ones that you think might be clinically relevant because of previous work that's been done. So in our group, we've started by doing a bit of all of them. We've done some whole genome sequencing, we've some measured whole exome sequencing, and we've some clinical exome sequencing, and we've also measured single nucleotide polymorphisms, which are genetic polymorphisms all across the genome. So the project is really only beginning. It's all under analysis at the moment, but what I wanted to show you was the research participants and the samples that we have started to collect and the amount of data that we have. So we've got 6 whole genome sequences. For each whole genome sequence, we've got 3 billion base pairs. So that's a huge amount of data to understand and to analyze. But it's a very exciting time for all of us in genetics. And so hopefully you'll come next year and we'll have loads of results for you. But I guess this is just to show then our future type of analysis. The pipeline that we'll do is how we will look at this type of genetic variation. So this is a figure from a recent paper where they looked at the variance within the human genome. So as I said, we've got 3 billion base pairs, but within those, only 3 million of them vary between all of us. And there's 3 million of those are common. But for autism, we also look at common and those that are rare and those that are de novo mutations. And that's illustrated by the 70 number there. So that's so the common ones are where they're within your mum and dad and the children. And then those that are rare are just very rare in the population and perhaps only in one parent. And then we look at the de novo types of mutations that happen spontaneously, so aren't in mum and dad, but are in the children. So these are the types of analyses that we're doing within our families, because there's evidence to suggest that all those different types of genetic variation are important for autism. And this is just a recent study by a collaborator where they have collected nearly 5,000 cases and controls, and they like us, they look at families, so they collect DNA samples from blood, samples from families, they collect cases and controls, so mum and dad, and cases and unaffected controls. They take out their DNA, they capture the exome, so they do DNA sequencing on the coding genes, and then their type of analysis that they do is they compare the type of rare mutations between cases and controls. And it's really interesting the type of results that they find this type of analysis can lead to revealing genetic variation linked with genes. So this is just a picture here to illustrate chromosome 2, one of our chromosomes, and along here are the lines illustrate the genes, and then the red dots illustrate where there's variation within these particular genes within cases, but not within controls. And then this is potentially areas of interest for follow-up for future research. And then, of course, we're also more interested in the protein coding genes. So in the genes, in the 20,000 genes, looking at variation within the genes and not just in the whole genome. And sorry, this just illustrates your 22 different chromosomes along here. Louise showed a very similar plot, and those that are up here, these are called Manhattan plots because they're like the Manhattan skyline. So you're looking for dots up the top there, and anything there is potentially really interesting in relation to understanding more about autism. So really, I suppose, what's next in autism genomics? That's why we're all here today, and I suppose part of our research is to identify high impact genetic loci, to understand the neurobiology and pathogenesis, and to really to personalize medicine. And collaboration is key in terms of in all areas of research, and we'll talk more about that later on. But from my point of view, from every stage of the research, in our group we collaborate a lot with work done in the US, in the UK, here in Ireland, and that's really key, so that because as you can see the amount of data that's there, the amount of samples that are required, but also collaboration in this setting is extremely key as well. And I suppose I wanted to highlight 3 things that I think that are important for the importance of, or the future for autism research. And this figure really caught my eye, actually. It came out a few weeks ago, and it just illustrates where the most research happens in the world, and the darker red colours illustrate the number of samples that take part in research. So you can see in the UK, Northern Ireland, in America, it's really high. There's a lot of research going on there. There's a lot of research participants take part there, but and it's wonderful, the results are fantastic, but the problem is that it's not representative of the whole world, because we, like Irish people, have different genomes than the UK, than America, so it's really important that we all have a place in the future of research. And and we'll talk more about that with Michael later on at lunchtime. And the second thing I thought for the future of autism genomics, this paper, a research paper, so you probably just to fill you in, that when we publish a study or do some research, we always write it up in a scientific journal and it gets published, and that's what we call being published. It's just writing your results up, and this one just came out yesterday in a really important genetics journal. And one thing that really caught my eye was an analysis they did, and this was in a study of 15,000 autism cases and controls, and they looked at 3 million or 500 million single genetic markers. And one of the analyses they did was they did a genetic correlation with autism and other phenotypes such as cognition, psychiatric phenotypes, education and personality. And shown here are the phenotypes where there was a significant genetic correlation with autism. So there's IQ, education and attainment, sleep, depression. So it just goes to show, I suppose, the importance of not just looking at autism, but also looking at other important factors related to autism and other important phenotypes. And of course, then the third thing that's important for following up in autism genomics is understanding the genome. I mean, we've talked about that there's 3 billion base pairs, and we really only understand a small amount of that. And a lot of work is going into understanding the bits of the genome that we don't know about. This is a picture of the ENCODE project over in the States, where they're trying to figure out, like the green bit here is just, the green bit is where the gene is, but there's all these important bits before and after the gene that are important for regulating how genes work, how proteins are made, how biological processes are working. So there's a lot more research to be done in figuring out that, and then what that could be potentially really important for understanding autism genomics. So that's it. Thank you very much for listening. Thanks to the research team.